Day 4 of explaining something very simply under 2 minutes so I can understand it better. Yesterday I spoke about cancer heterogeneity and its three causes: genetic, epigenetic, and microenvironmental. Today I will talk about genetic heterogeneity in detail. So, the first reason of genetic heterogeneity is point mutations. So every time a cell divides, it has to copy its DNA, and during this process, errors can occur. For example, deletion of a letter, addition of a letter, substitution of a letter. And with the constant turnover of tumor cells and the large tumor cell populations, some of these mutations will affect genes with known cancer relevance, like tumor suppressor genes. The second reason of genetic heterogeneity is aneuploidy. Aneuploidy is when a chromosome number is either more than 2 or less than 2. If it's more than 2, it's trisomy. If it's less than 2, it's monosomy. And it's not. The third reason of genetic heterogeneity is polyploidy, also can be called WGD, whole genome doubling. And this is also where aneuploidy arises. So in aneuploidy, one chromosome is more in number or less in number. But in polyploidy, the entire set of chromosomes, all the chromosomes are more in number. For example, all the chromosomes, instead of 2, there are 3 copies or 4 copies, tripled or quadrupled. And this is not very stable. So some of these copies disintegrate, and this is where aneuploidy is born. The fourth reason is chromosomal instability, which is an increased rate of genomic mutational errors involving loss, gains, or translocations of large fragments of DNA. Now, here what happens? Chromosomal instability allows a much faster rate of genomic diversification. This helps speed up the process of evolution during tumor development and cancer therapies. So cancer cells with the wrong number of chromosomes mutate much more often than with normal number of chromosomes. CIN therefore is a self-amplifying process. If there is very less chromosomal instability, the tumor cannot evolve fast enough to resist therapy. If there is too much chromosomal instability, the tumor itself is too unstable and dies. But if there's intermediate level of chromosomal instability, that is the maximum danger point. It's just enough diversity to escape treatment and enough stability to keep growing.